decreased. And importantly, what I want to draw your attention to is that the PAR2 deficiency attenuates the loss of vascular smooth muscle cells. So if you look at alpha-actin smooth muscle cell stain, you can see that PAR2 deficiency has a dramatic, I'm not going to say increase, but a dramatic reduction in the loss of smooth muscle cell staining. So when we take this out for a long-term study, 24 weeks, again in the LDL model, I'm not going to show you too much of the data, but because it's already published, but basically, again, similar reductions in atherosclerosis. Again, though, I want to draw your attention to the PAR2 or the smooth muscle cell actin. And when you look in terms of the PAR2 proficient on top and the deficient on the bottom, again, you can see that the smooth muscle cell actin is still highly present, even at 24 weeks of atherosclerotic feeding. Going into a little bit more detail, but again, don't want to get too much into the weeds because this is published. You can see that, you know, CD68, necrotic core, all down. But again, alpha actin lesion composition is up. And if you look at the fibrous cap composition, smooth muscle cell alpha actin is also way up. So we wanted to ask, are these effects dependent on hematopoietic and non-hematopoietic? And so again, standard bone marrow transplantation study, giving us 4 chimeras, feeding a Western diet for 12 weeks. And what you can see is that basically everything with PAR2 recipients has a decrease in atherosclerosis, either with the sinus as well as in the lesion. And so this points to a non-hematopoietic source of atherosclerosis. Again, if you look at macrophage deposition is all related to non-hematopoietic markers. But again, I want to draw your attention to alpha-actin smooth muscle cell, which again is does not have as much of a reduction as with the non-hematopoietic or the hematopoietic as well as the wild type control. So just to summarize this published data, PAR2 deficiency attenuates diet-induced athero at 12 and 24 weeks, attenuated macrophage accumulation, importantly augmented smooth muscle cell presence. It appears that non-hematopoietic PAR2 deficiency recapitulates the effects from the whole body. And I didn't show it, but there's a lot more data related to how PAR2 deficient mice have attenuated production of cytokines and chemokines specifically from smooth muscle. So now I kind of want to go into what we think is the mechanism behind what's happening here. And this is all unpublished so far. I saw Mete on here earlier, so thanks for coming, Mete. In collaboration with him and UCLA, we basically wanted to look and see, you know, what's going on with PAR2. What exactly does it correlate with in the hybrid mouse diversity panel? And one of the biggest hits that we had was KLF4. And so if you look at the R value and the P value, it was highly significant in terms of all of our other hits. And so, you know, we wanted to kind of verify this and see what was going on. And so if you look at PAR2 deficient cells as well as just wild type smooth muscle cells, if you treat them with a PAR2 agonist, you can see that KLF4 expression just goes up through the roof. And then obviously PAR2 deficiency with a PAR2 agonist doesn't result in, you know, any production of KLF4. So I'm sure most of you are familiar with the studies by Gary Owens and Olga Cherepanova and everyone else related to how KLF4 is critical for the modulation of smooth muscle cells into a macrophage-like state. Obviously, this was first put forward by Ed Fisher's lab back in 2003, where basically he showed that if you treat smooth muscle cells with a cyclodextrin cholesterol, what happens is you start to get less smooth muscle cell markers, less alpha actin, less tropomyosin, and more macrophage-like markers, high expressing CD68, MAC2, and all of these can obviously be quantified with relative abundance of mRNA, and a lot of different markers. So what we wanted to do basically was look and see that is there something going on with PAR2 and KLF4 on this front. And what we did is we took PAR2 proficient and deficient smooth muscle cells, treated them with the cyclodextrin cholesterol for 72 hours, and then quantified and saw what was going on. Strikingly, what you can see is that, and I'll blow this up here in a minute, you can see that the PAR2 deficient smooth muscle cells actually don't really accumulate any lipid whatsoever, which is extraordinarily interesting. If you go back and you start looking at the actual quantitative markers, you can see that PAR2 deficient smooth muscle cells, when treated with this cholesterol, does not lose its alpha actin or its MHC, and in fact does not wind up gaining CD68 and MAC2. So it doesn't appear to be going through this transdifferentiation process. And again, using Wrong and Ed Fisher's paper, we looked and saw, is there, in terms of macrophage-like state, what's going on with beat uptake? And you can see that when we, again, take cholesterol, 72-hour treated cholesterol, PAR2 proficient, deficient smooth muscle cells, and then we throw on beads for 24 hours, you can see that basically the bead accumulation in the PAR2 deficient animals is much, much less than in the wild type animals. And this is quantified here, where you can see obviously latex beads per cell around 8 versus 2 to practically nothing in the PAR2 deficient animals. So it looks like PAR2 and KLF4 are actually highly associated, and that PAR2 deficiency attenuates vascular smooth muscle cell to macrophage transdifferentiation under cholesterol pressure. So I'm not going to go into too much more of the detail because, again, we're still working on it, and unfortunately, we were getting really hot and heavy with this before COVID shut us down. So we don't have a whole lot more to say about the PAR2 KLF4 story. But something that we were working on before we even found KLF4 was a connection between PAR2 and QR. My collaborator down the hall, Mike Trainer, is a huge QR expert. He works on the role of QR as it pertains to cardiac hypertrophy and MI. And so we tried to start linking our two studies together, or our two labs together, and we found a connection between QR and PAR2. So QR is basically it binds to AU-rich regions in target mRNA and usually mediates stabilization, but can also lead to degradation in certain situations. Most of what you know about QR is primarily in cancer biology, but there have been a few studies lately that have shown effects of QR in cardiovascular disease as well as in atherosclerosis. And so interestingly enough, the first thing we did was we looked and saw whether or not PAR2 has any of these QR-rich AU binding regions. And in fact, we saw that PAR2 has 7 of these AU-rich binding regions, which according to Mike is one of the biggest or one of the most proliferative regions he's seen in a protein. So interestingly enough, it looks like PAR2 and QR were made for each other. And so if you look at basic stabilization studies using actinomycin D, as well as using a QR inhibitor, you can see that if you take smooth muscle cells and you treat with actinomycin D, and then also give a QR inhibitor on board, which is represented in blue, you can see that PAR2 mRNA completely starts degrading when you take QR away. And this is quantified over here at 3 hours. So you can see basically it looks like QR is stabilizing PAR2 mRNA pretty robustly. And so this is just a basal level of QR in PAR2 deficient smooth muscle cells. We wouldn't expect to find anything, but lo and behold, if you just take smooth muscle cells from a wild type and a PAR2 deficient animal, and you look at basal QR mRNA, it's already significantly downregulated in these PAR2 deficient smooth muscle cells. So there's definitely a link between QR stabilizing PAR2, but it also looks like there might be a feedback loop from PAR2 to QR. And so if you actually look at PAR2, again, PAR2 proficient and deficient smooth muscle cells treated with either cholesterol or just wild type you know, solution, you can see that if you stain for HUR, and HUR is mainly localized inside of the nucleus until it's activated and then it shuttles out, you can see that HUR in the control stays in the nucleus, and if you merge it together, you get this nice aqua color. But in a wild type smooth muscle cell, if you treat with cholesterol for 72 hours, the HUR all shuttles out and becomes activated. So it's all in the cytoplasm. If you look at the PAR2 vascular smooth muscle cells, and specifically at the cholesterol treated, again, you can see it's mainly, again, sitting in the nucleus. It doesn't shuttle out and it doesn't become activated. So again, there's some role between PAR2 and HUR in terms of cholesterol activation. And this is just a blown up picture because obviously looking at 16 small panels can get a little bit dizzying. So again, you can see that when you're looking at the wild type vascular smooth muscle cells versus cholesterol treated, you can see that the cholesterol again shoves all the HUR out into the cytoplasm, whereas PAR2 vascular smooth muscle cells don't have any shuttling at all. And so this is kind of our mechanism that we think is happening here. We think that PAR2 activation leads to the activation of KLF4, but we also think that PAR2 leads to the activation of HUR. HUR then shuttles from the nucleus out to the cytoplasm, where it then stabilizes PAR2 mRNA and feeds back to basically increase the amount of PAR2. Now, we're currently looking to see whether or not HUR actually stabilizes KLF4 too. Because like I said, we've obviously shown that PAR2 and KLF4 are also linked. And so there looks like there's a very redundant system built in here where PAR2 is creating an increase in KLF4 and HUR. HUR is then feeding back on PAR2 and KLF4, leading to all of these different changes, inflammatory cytokines, and importantly, the vascular smooth muscle cell phenotypic modulation to macrophages. And so ongoing studies that we have in the lab is we're confirming our non-hematopoietic origin with PAR2 flox mice and a My11 Cree promoter. We've successfully obtained and are breeding and almost done after 2 years of breeding. Finally have the PAR2 floxed My11 Cree Rosa flox stop EYFP stop ApoE reporter. So hopefully we will be able to tell whether or not smooth muscle cells that originated are actually turning into macrophages in the lesion sometime soon. And then ongoing studies define the role of HUR. We also have the HUR flox mice on a My11 Cree background. So we're getting there slowly but surely. So next I kind of want to talk about the role of PAR1 and PAR4. So like I said, over here you can see that PAR1 and PAR4, PAR1 is also highly upregulated in carotid plaques, but PAR4 isn't. Now this is interesting because PAR4 itself doesn't actually, PAR4 itself is just a signaling receptor. The activation receptor is actually PAR3. And so if you talk to a lot of experts in the field, and this is changing, they actually think PAR3 might be able to signal on its own in certain capacities. But for the most part, PAR4 needs PAR3 to signal. And so the fact that PAR4 is unchanged here doesn't mean that it's bad. Also just means that PAR3 is the actual thrombin activation receptor. So this is going to get a little dicey. It's always hard to explain that there's differences in humans and mice. But platelets are obviously critical for hemostasis in the coag cascade. In terms of mice and humans, human platelets have PAR1 and PAR4 on their surface. PAR1 is the main activation receptor. It activates with very low concentrations of thrombin. PAR4 also activates with thrombin, but again, you have to have higher concentrations and you need PAR3 to signal through PAR4. In terms of mouse platelets do not have PAR1. Mouse platelets only have PAR3 and PAR4. And so this is important when you're talking about the correlation between human studies and mouse studies. When you're talking about platelets, in humans, you're looking at PAR1, and mice, you're looking at PAR4. And so you got to think about, that kind of cross when you're thinking between humans and mice. And so the literature, basically going back to 2006, if you look at global thrombin inhibition with a direct thrombin inhibitor, it reduces atherosclerosis in an ApoE model. And this, I believe, 2006. And then if you go a little bit forward to Borisov, you can see that, again, targeting prothrombin reduces atherosclerosis in an ApoE model. And so, we wanted to look and see what was the effect of PAR1 on atherosclerosis. Again, just taking PAR1 proficient, deficient on an LDL background, Western diet for 12 weeks. And we didn't find any difference, oddly enough. We thought we might, but no difference. Again, PAR1 is not on platelets and mice, so it means that this would be PAR1 on all of the ancillary cells, smooth muscle cells, macrophages, and everything else. So no effect with PAR1. Interestingly enough, PAR4 had already been examined by Sean Coughlin's lab back in 2009. And what they found is a PAR4 proficient on an ApoE deficient model fed a Western diet, the deficiency had no effect. And so they concluded that thrombin-mediated platelet activation was not required for the development of athero in an ApoE model. Interestingly enough, though, a few years later, there was a trial that came out saying that PAR1 inhibitors decreased all-cause mortality. So it was kind of odd that you would have an effect of a PAR1 inhibitor in a human, but no effect of a PAR4 deficient animal and a mouse. And so we decided, what the heck, let's take a look at this. Let's look at a different model. Let's look at LDL. And so we bred PAR4 proficient deficient animals on an LDL, high-fat diet for 12 weeks, quantified athero, and lo and behold, PAR4 deficient animals have a significant decrease in the lesion area in the aortic sinus, and as well as the lesion area in the aortic arch. And so looking at the chimeric study, again, PAR4 is the main thrombin receptor on platelets. So, going into this, we figured it was a platelet-mediated effect. And, it turns out that it is a hematopoietic effect. Now, we assume it's platelets, but, there's nothing saying that it's platelets until we get some kind of a flox mouse, which I don't believe exists yet for PAR4. So we kind of wanted to look. Obviously, you have PAR4, you have PAR1. They can both be activated by thrombin, right? So we wanted to see if you take dabigatran, which is a thrombin inhibitor, and you throw it into PAR4 deficient animals, do you have a significant attenuation of your reduction of athero, right? And so dabigatran, again, is a thrombin inhibitor, and it basically decreases the production of fibrin and platelet activation. And so when we threw in dabigatran here in PAR4 deficient animals, you can see that dabigatran or placebo had no additional effect of reducing atherosclerosis versus just the PAR4 genetic deletion alone. You can also see that if you take a wild type animal and you give dabigatran, that it reduces atherosclerosis to the level of PAR4 deficient and PAR4 deficient with dabigatran. So this basically says that thrombin mediation of atherosclerosis seems all to be dependent on PAR4. So basically, to summarize the PARs all the way down, right? PAR1 had no effect. PAR2 attenuates athero via non-hematopoietic, and PAR4 attenuates athero via hematopoietic mechanism. Now, this is kind of where we got into a little bit of conflict, right? Because very soon after this was published, a couple other studies started to be published. And so right after our study was published, a study was published that said PAR2 deficiency attenuates atherosclerosis via hematopoietic cells. We found non-hematopoietic cells. Another study was published same year that PAR1 deficiency attenuates atherosclerosis. And obviously, I've already shown you that PAR4 deficiency has no effect on atherosclerosis. And all of these things in common are the ApoE model. We've done everything on the LDL model. All of these are on the ApoE model. And so, we get back to this old Godfrey Getz, kind of grudge about the difference between the ApoE and the LDL model. And so looking it up, you can actually see that ApoE does have a role in platelet aggregation. And it's very ill-defined in the literature, but ApoE protects against platelet aggregation in some respects. So if you take it away, you get more platelet aggregation. At least that's the theory, I suppose, in the literature, although I've never found anything to back that up. So PAR 2 and PAR 1, I don't think there's much I can do to try to figure out what's going on between ApoE and LDL there. But with platelets, I can at least do something quantitative and try to figure out what's going on here, right? So we started to look at the difference between ApoE and LDL in terms of platelet reactivity. First, we used the ferric chloride model of thrombosis. And basically, this model, you put a patch of ferric chloride onto the carotid artery of a mouse. And then down here, you use a transducer to look at blood flow. And what happens is, blood will start clotting off because platelets start accumulating and blood flow will start reducing. And so what we do is we measure time to occlusion, or time that the blood flow reaches basically a minimal point or zero. And what you can see in our published studies from 2012, if you take LDL receptor deficient animals and you give them high-fat diet, you get a significant reduction in the time to occlusion. So they clot faster, basically. So we wanted to take a look and see what's the difference between ApoE and LDL in this clotting model. Do platelets accumulate faster, and do they clot faster? So we took we took 6 weeks old mice. Obviously, ApoE are spontaneously hypercholesterolemic, so it's impossible to get a non-cholesterol mouse with ApoE. So we tried to take as early of a mouse as we could to try to mitigate any kind of atherolesions that might form. So we took 6-week-old mouse, and we also looked at Western diet starting at 4 weeks of age up to 6 weeks. And then we took the traditional 8 weeks plus 8-week-old mice plus 8 weeks of Western diet. And what you can see, interestingly enough, again, with ApoE in red and LDLR in blue, you can see that ApoE deficient animals, whether in the 6-week-old mice and the 6-week-old with 2-week-old Western, or the 16-week-old mice with 8 weeks of Western, you can see that ApoE reduces the time to occlusion in all 3 situations. So obviously, these guys are clotting faster, and that leads us to what's going on with the platelets. So looking at traditional aggregometry, we found that, and traditional aggregometry for those who aren't proficient in coag assays, what happens is you take platelet-rich plasma, you throw it in the tube, and it's very cloudy. When you make the platelets, basically, when you stimulate the platelets, they start clumping together, they fall to the bottom, and then light is able to transmit through. And so you go from a transmissibility of 0 all the way up to a transmissibility of, close to 90%. And what you can see is an ApoE deficient animal fed a Western diet for 8 weeks, or an ApoE deficient animal with chow versus an LDL receptor deficient animal on Western or an LDL on chow. You can see that ApoE has an increase in platelet reactivity, basically, on Western diet and on chow, much greater than the LDL receptor deficient animal. And so using a couple different assays, and I'm going to kind of speed up so I can get through here. A couple different assays. We used impedance aggregometry, which basically uses an electrode and causes platelet aggregation. You can see that with ApoE deficient animals in red, you can see that there was, in terms of, well, I'll just go with the LDL. you can see that the LDL receptor deficient animals had a decreased aggregation velocity, decreased area under the curve, and remarkably, and we hadn't looked at this before, they actually have decreased platelet counts basally. And so some of the experiments that I really wish we would have gotten to before COVID hit are these really cool microfluidics experiments where basically what you would do is you would throw ApoE whole blood in here, LDLR whole blood in here, and then it flows through this microfluidics channel, and across this little stretch right here, there's this stretch of collagen, and then the platelets start binding to the collagen. So it's like a little mini microfluidic clotting model. So that's going to kind of verify the ferric chloride thrombosis model for us. So wrapping this all up, we're aiming for a par 4 athro paper with the inclusion of the ApoE and LDL explaining the difference between our paper and Coughlin's paper sometime in the next few months if we can get back into the lab and start cranking this out. So conclusions and takeaways, basically, the role of PARS in athro is complex to say the least. My 2 cents, I think part 2 has a role in smooth muscle cell mediated athero progression and macrophage transdifferentiation into macrophages. But again, another study says maybe that's not the case. So that's my 2 cents. And then obviously a caution, right, is what we're finding is there are definitely differences in terms of platelets with the LDL and the ApoE model. So if you're looking at something that is associated with platelets, you might want to take caution using the ApoE model because you might not get something that is not falsely elevated, let's just say. So with that, obviously I'd like to thank my lab, the many members of it, and my sources of funding, all the collaborators. Collaborators are what makes this run. And then thank you for your attention. Happy to take any questions.
That was awesome. Thank you, Phil. So I'm just going to ask any of the participants who might have questions to raise their hand. And while they do, I'm going to ask Phil a question. So something that struck me in your part 2 data was that it would seem to me almost that part 2 is acting almost like a lipoprotein receptor. Do you think it didn't look based on your cartoon of its, a structure necessarily that it's a active, I don't know, what is it? Like, would it be actually taking up lipoproteins, or do you think it's facilitating potentially lipoprotein uptake, or what it's doing? Because why are those cells so different in terms of their cholesterol accumulation when you incubate with excess cholesterol?
So that's a great question. We have basic hints about what's going on, but we're still trying to work through it. But this kind of works back a little bit to the MIR33 story that we're working with you and Catherine and even Nick out there in California on a little bit. So what we think is happening, honestly, is that PAR2 might be cooperating with ABCA1, and it looks like there's a role for tissue factor mediated regulation of MIR33 as well. So the cartoon would look insane if I drew it all out. But what we think is going on is that PAR2 is sitting right in the middle. It is, for all intents and purposes, the tissue factor receptor. And it is mediating everything from lipid efflux to inflammation and so many other things. But I don't think it has a direct role. I think its role is indirect as a co-activator of these different receptors.
Very cool. Okay, Ada, you've got a question. Whoops. You unmuted there, Ada?
Can you hear me?
Okay, yeah.
Thanks, that was awesome. I have actually 2 questions. So one is regarding your last part of the talk. I was wondering if you compared your LDL receptors to wild types. Do LDL receptors aggregate as well as wild types?
So that's an interesting question. So far as we can see, yeah, they have no difference between a wild type C57 and an LDL receptor. It's when you jump up to the ApoE that you start getting differences.
And then my second question is regarding your part 2 story. And I was wondering, you showed some microscopy, and I saw that the smooth muscle cells that are part 2 knockouts were actually, they had smaller nuclei. So I was wondering morphologically if the smooth muscle cells look the same and act the same other than cholesterol uptake and what you see in vivo.
So that's a great point. I don't think I've ever noticed that. So thanks for pointing that out. I'll have to go back and look at that. As far as we can see, they don't have any basal physiological differences, versus wild type cells. But it's apparent that there are definitely issues when it comes to inflammation. So if you trigger them, they have less of an inflammatory response, and they have less of a, basically, if you want to call it a fight or flight response, they have less of that activation potential. But in terms of basal function, motility, spreading, as far as we haven't noticed any differences.
Thank you.
Great. Okay, is there anyone else that has questions? Please raise your hand if you do. And yes, going once, going twice. No. Okay, well, thank you very, very much, Phil. That was awesome.
Thanks, Katie.
So next up, actually, we've got 2 speakers, and Ebru, I think you're going to take it from here.
Yeah, thank you, Katie. Thank you, Phil. Great work. I want to introduce 2 speakers from, they're from Thomas Paul Nichols' lab in Dalhousie University. And I'm really proud because 2 trainees will be talking today from the same lab. And we really want to encourage trainee talks in the future months of these seminars. So Thomas, I invite you to introduce the great trainees from your lab, if you're here.
Thank you, Ebru. Hello, everyone. And thank you, Cardiometabolism Zoom seminar series for this opportunity to our lab. I'm very pleased to introduce 2 individuals from my lab, Dr. Deepshika Biswas and Ms. Purvi Trivedi. Dr. Biswas did her PhD from IICB India on examining the role of ATGL, or adipose triglyceride lipase, in muscle proliferation and also in hepatocellular carcinoma. She then joined my lab as a postdoctoral fellow in May of 2017, where she started working on branch-chain amino acid metabolism and how it impacts musculoskeletal health and disease. And so she's sharing some of the clinical data that we have obtained on branch-chain amino acid metabolizing enzymes today in the last few years, and how that enzymes play a role in cardiac surgery outcomes that is being described in this talk. Over to you, Deepshika.
Okay, can you see the slides? Right.
Yes.
Okay. So thank you for the introduction, Thomas. I would also like to thank the organizers for giving me this fantastic opportunity to present my work today. And as Thomas mentioned, I'll be talking about branch-chain amino acid catabolism in the heart. So branch-chain amino acids, or BCAs, are comprised of the 3 essential amino acids, leucine, isoleucine, and valine. And they have been attributed with diverse physiological roles. For example, like here, that I'm mentioning over here is like maintaining the balance between muscle protein synthesis and breakdown, as well as modulating glucose and fatty acid metabolism in various tissues. Now, branch-chain amino acids can enter into the circulation when they are released from proteins, either from the diet or from other tissues, and they can be cleared from the circulation when they get reincorporated into new proteins, while the remaining BCAs in circulation can get oxidized in various tissues. Now, although the BCAs have these beneficial effects under negative energy conditions, chronic increases in circulating BCAAs have emerged as a potential biomarker for several obesity-associated metabolic diseases over the past decade. Indeed, increased plasma BCA levels have been associated with obesity and insulin resistance in humans, Zucker fatty rats, diet-induced obese rats, as well as leptin-deficient OB mice. So this has led to the hypothesis that the catabolic defects of this BCA pathway could result in this increased accumulation of circulating BCAAs that is observed in type 2 diabetes and associated comorbidities like cardiovascular diseases. So to give a brief overview of the BCA catabolic pathway, BCAAs are reversibly transaminated to form branched-chain ketoacids or BCKAs by the enzyme branched-chain aminotransferase or BCAT. The fate of these BCKAs then depend on the activity status of the enzyme supercomplex called branched-chain ketoacid dehydrogenase or BCKDH. BCKDH can be inactivated by phosphorylation by a kinase called BCKDK that does not allow the downstream oxidation of the branched-chain ketoacids. On the other hand, the phosphatase PPM1K can dephosphorylate the supercomplex and activate it, allowing the oxidation of branched-chain ketoacids by various distal BCA catabolizing enzymes, resulting in the formation of the three main acyl-CoA species, acetoacetyl-CoA, acetyl-CoA, and methylmalonyl-CoA, which can in turn act as intermediates in various metabolic pathways. Now, several studies over the past few years have identified various pathological stress signals that can impair the BCA catabolism in different tissues that can ultimately lead to increases in circulating BCA levels. So we were interested to investigate the impact of obesity on BCA catabolism in the heart. We found that in mice fed high-fat, high-sucrose diet for 16 weeks, there was a significant reduction in both the proximal as well as the distal BCA catabolic genes within the heart. Further, we also found that high-fat, high-sucrose significantly reduced the KLF15 mRNA levels in the heart, and KLF15 is an important transcriptional regulator of this catabolic pathway. We also found that this reduced BCA catabolism within the heart was resulted in increased serum accumulation of branched-chain ketoacids, which indicated that the BCA catabolism in the heart can be suppressed in response to an obesogenic milieu. Now, our laboratory is part of an OPOS clinical trial, which involves patients with obesity that have enrolled at the St. John Regional Hospital to undergo elective cardiac surgery. So we could procure the atrial appendage and the subcutaneous adipose tissue surrounding the heart from these patients during surgery. And for the purposes of our study, we stratified these patients into different classes of obesity based on their BMI. We found that the enzyme activity of BCKDH, which is also the rate-limiting enzyme of the pathway, was significantly reduced in the adipose tissue of severely obese patients. Consistently, we also observed increased protein expression of the kinase BCKDK and corresponding increases in phosphorylated form of BCKDH, which is the inactive form. We also found reduced protein expression of the transcription factor KLF15, which is within the obese adipose tissue. Further, we found that this decreased BCA catabolism within the adipose tissue was also significantly correlated with increased length of stay in the hospital following surgery, which suggested that the impaired BCA catabolic gene expression within the adipose can influence post-operative outcomes. Now, after we found these changes within the adipose tissues, we were interested to see what happens within the atrial appendage of these patients. And we found that the BCA levels within the atrial appendage was significantly upregulated in the obese patients. However, we did not find any significant reduction in the enzyme activity of BCKDH within the atrial appendage, which also explained a reduced ratio of ketoacid to amino acid within these patients. Now, further, this reduced BCKA to BCA ratio was again significantly correlated with increased BMI, as well as increased length of stay in the hospital following surgery. So together we theorize that the reduced BCA catabolism within the subcutaneous adipose tissue might result in shunting of the excess BCAs towards the heart for oxidation. However, excess BCA entry into the atrial appendage cannot be effectively catabolized, as we did not find any compensatory increase or any other changes in the enzymes within the heart, and that could contribute to the adverse post-operative outcomes that we saw. Now, our data is in agreement with previous studies which demonstrated impaired cardiac function in mouse models of BCA catabolic defects. And also these studies also reported increased accumulation of branched-chain ketoacids as well. Now, further, many studies have now directly reported the role of the branched-chain ketoacids in direct impairment of mitochondrial respiration, compromising glucose uptake, and increasing oxidative stress within the cardiomyocytes. So we therefore hypothesize that at least some of these metabolic complications which are mediated by the branched-chain ketoacids could be explained by its effect on cardiac insulin signaling. So we examined insulin signaling in response to acute exposure of individual ketoacids in cardiomyocytes isolated from neonatal rats or adult rats. And we found that pre-incubating these cardiomyocytes with ketoleucin could significantly reduce insulin-induced AKT phosphorylation at serine 473, and it also significantly impaired the AKT activity. Now, this was also true for the other ketoacids derived from isoleucine and valine. So together, our study suggests that treatment with the exogenous BCKAs can impair insulin signaling in the cardiomyocytes. We next wanted to investigate whether altering the endogenous BCKA levels by genetic modulation of BCKDH could influence insulin signaling in the cardiomyocytes. So for this, we targeted the BCKDH-A subunit of the supercomplex, and we found that silencing BCKDH-A robustly increased intracellular accumulation of branched-chain ketoacids, which could then impair insulin-induced AKT phosphorylation in the cardiomyocytes, which is also what we saw with the exogenous treatment with BCKAs. Conversely, we found that overexpressing BCKDH resulted in a modest but significant decrease in intracellular BCKA accumulation that could potentiate insulin-induced AKT phosphorylation. So together, our data suggests that altering or affecting the clearance of BCKAs by genetic modulation of BCKDH-A can reciprocally regulate insulin signaling in the cardiomyocytes. So we next wanted to examine whether the effect of BCKAs on insulin signaling pathway is associated with an increased activation of mTOR, because it is known to negatively regulate insulin signaling. We found that activating phosphorylation of mTORC1 and its downstream target P70S6K was significantly increased upon exogenous ketoleucin treatment. And we further found that depleting BCKDH levels, which also resulted in that increased accumulation of BCKA, could also increase phosphorylation on both mTORC1 as well as P70S6 kinase, suggesting that BCKA-mediated insulin signaling impairment could be attributed to the increased mTOR activation. So to summarize our findings, we show that the BCA catabolism in the subcutaneous adipose tissue and atrial appendage is altered in obese patients undergoing cardiac surgery, and this altered BCA catabolism also correlated with adverse postoperative outcomes. We also show that diet-induced obesity suppresses the expression of BCA catabolic enzymes in the heart, which could influence the systemic levels of branched-chain ketoacids. Further, we found that acute exposure of individual BCKAs can directly inhibit insulin-induced AKT phosphorylation in the cardiomyocytes, and altering the intracellular BCKA levels by either overexpressing or silencing BCKDHA can reciprocally regulate this insulin signaling. And finally, we think that the BCKA-mediated activation of mTOR signaling can play a role in its inhibition of insulin signaling in the cardiomyocytes. So I would like to end by acknowledging my supervisor, Dr. Thomas Pullinel-Kunnel, our collaborator, Dr. Petra Kinnisberger, all our lab members, and the IMPART team for the clinical samples and the funding agencies. Thank you.
Thank you. And Thomas, do you want to introduce Purvi?
Yes, thank you, Ebru, once again, and I'm very pleased to introduce Ms. Purvi Trivedi, who is a doctoral trainee in the lab. Purvi came to my lab after completing a master's in pharmacology in 2013, and she since then completed her PhD, master's in my lab, examining the role of different nutrients on myocardial autophagy. And in her doctoral program, Purvi has extended this work further by examining the role of transcription factor EB, the master regulator of lysosomal function in cardiomyocyte health and disease. And today she will present a few pieces of data showing the phenotypic characterization of cardiac specific TFEB knockout mouse. Over to you, Purvi.
Thank you, Thomas, for the introduction. So as Thomas suggests, today I'll be talking about roles of transcription factor EB in the heart, especially in the context of obesity and diabetes. So just with the brief background, our lab actually focuses on examining the substrate utilization in the heart. And impairment in this substrate utilization is one of the proposed mechanisms used to understand the obesity and diabetes related cardiovascular complications. So how are these substrate being first utilized in obese and diabetic heart? So for example, during obesity and diabetes, there is a decrease in glucose utilization, which can lead to hyperglycemia and glucotoxicity. Simultaneously, there is also a increase in fatty acid utilization, which can cause steatosis and lipotoxicity. And together, this condition can cause glucolipotoxic stress within the heart. And furthermore, this glucolipotoxic stress in the heart can cause endoplasmic reticulum stress and mitochondrial dysfunction, as well as recently we demonstrated that glucolipotoxicity impairs lysosomal autophagy and function, and that can likely cause a cardiomyocyte injury and heart failure. So before we actually go further and talk about proteotoxicity, we first need to understand how the proteostasis takes place in normal circumstances, such as in physiology. So basically proteostasis, there are two main pathways responsible to ensure proteostasis. One is ubiquitin proteasomal pathway, which is not the focus of my talk for today. And second is proteolysis by lysosomal autophagy. So lysosomal autophagy is activated in response to stress and starvation. And once it is activated, it recycles unwanted materials such as toxic proteins or damaged organelles to acquire nutrients. And then these nutrients can further be used as a source of energy to ensure cell survival or cellular homeostasis. Now, on the other hand, lysosomal autophagy can be inhibited in response to nutrient abundance as well as in certain pathological conditions. So basically, there are three different forms of autophagy described. For those are macroautophagy, chaperone-mediated autophagy, and microautophagy. And since any forms of autophagy eventually culminates into lysosome, so not only the proteins involved in autophagy, but the number of lysosome and the activity of lysosome, as well as the number of lysosome and the activity of lysosome is important to ensure the proper activation of autophagy and the cellular survival. So, and then most importantly, how are these processes regulated in the cells, such as the proteins involved in autophagy or the lysosome biogenesis and function? And indeed, in 2009, Andrea Balabio and group, as well as other researchers, have identified a protein called transcription factor EB, which is called TFEB. TFEB is a member of MITEF family, which also involves other members such as TFE3, MITEF, and TFEC. And as name suggests, it's a transcription factor. It's a basic helix loop helic leucine zipper factor, and it's a nuclear resident protein. And again, it can be activated in response to starvation and stress, and as well as it can be inhibited in response to through its phosphorylation via mTOR and ERK. So once it is activated, it can induce the transcription of genes, which is involved in various biological processes and which involves, most importantly, such as lysosome biogenesis function and autophagy. as well as TFAB can also involve in regulating some of the processes, which is cellular survival, DNA damage, and repair. And most importantly, it has also gained a lot of attention for its regulation on cellular metabolism, which involves mitochondrial biogenesis, lipid metabolism, and glucose homeostasis. So then we ask a question that does glucolipotoxicity negatively regulates TFAB to alter proteins involved in lysosomal autophagy as well as lysosomal function and lysosomal content to induce a cardiac injury? And to answer this question, we first wanted to examine the levels of TFAB in obesogenic environment in humans as well as in rodent models of diet-induced obesity. And like Deepshika suggests in her talk, that we were part of a OPUS clinical trial, which involves patients who had undergone cardiac surgery. So we categorize these patients as per their BMI into this category, which is non-obese, pre-obese, and class 1 obese. And then we procured the atrial appendage sample of these patients. And we first measured the TFAB protein expression in atrial appendage. And we found that TFAB protein levels were significantly decreased in obese class 1 patients compared to pre-obese and non-obese patients. And most importantly, we also found that there was a negative correlation between TFAB levels and BMI of the patients, meaning that high BMI, patients with high BMI exhibited low levels of TFAB in their atrial appendage. And we further recapitulate our clinical data in rodent model of diet-induced obesity, where we procured C57 black 6J mice, and then we fed these mice with high-fat, high-sucrose diet for 16 weeks. And we first examined the protein expression of TFAB. So in the heart of mice who were fed high-fat, high-sucrose diet, we found that TFAB nuclear content was significantly decreased. On the other hand, we found that phospho-TFAB, which is an inhibitory form of TFAB, was significantly increased, as well as the stoichiometric ratio of phospho to TFAB were significantly increased. And most importantly, we also measured the cathepsin activity to determine the lysosomal proteolytic function. And we found that cathepsin B activity were significantly suppressed in high-fat diet-fed mouse heart compared to that chaw-fed mouse heart. So together, we demonstrated that in obese diabetic heart, there is an increase in phosphorylated or inhibitory form of TFAB, which led to decrease in nuclear content of TFAB and likely causing lysosomal dysfunction and cardiomyocyte injury. So in our next experiment, we definitely wanted to see that if we are able to restore the TFAB levels back in the nucleus, is it able to reverse the pathology such as lysosomal dysfunction and cardiomyocyte injury? And to do that, we employed adenoviruses, which overexpresses human TFAB which is mutated at serine 142 to alanine. And this will result in the phosphorylation resistance of TFAB and lead to the increase in constitutive activation of TFAB in the nucleus. So we use neonatal rat cardiomyocyte, and then we infected these cells with adenoviruses, and from this immunostaining, we found that we were successfully able to see the increase in nuclear translocation of TFAB, and subsequently to adenoviral transduction, we further treated these cells with different substrates such as BSA, high concentration of glucose, palmitate alone, and a combination of glucose palmitate. And first thing we wanted to, we examined the caspase-3 activity which is a marker for cell death. And we found that in CMV-GFP control group, caspase-3 activity was significantly increased in response to palmitate as well as glucose palmitate treatment. And glucose palmitate, we used a combination of this treatment to simulate the glucolipotoxic stress in vivo. And we also found that when we overexpress TFAB, it is able to significantly reduce the caspase-3 activity compared to its CMV-GFP control group. And furthermore, we next measure the cathepsin B activity to assess the lysosomal function, and we found that cathepsin B activity were significantly suppressed in response to palmitate and glucose palmitate, as well as it was able to reverse the suppression in activity when TFEB levels were restored in the cardiomyocyte compared to its CMBGFP group, control group. So to summarize the first part, we found that TFEB play an important role in regulating lysosome biogenesis and function, as well as it is also important for cardiomyocyte cell survival in the heart. And once we know that TFEB is important in regulating certain biological processes, we were curious to know what other pathways have been regulated when TFEB is lost in the heart, especially in the condition of obesity and diabetes. And to examine that, we developed a cardiomyocyte specific constitutive TFEB knockout mouse model. And this is just the confirmation that we were able to delete TFEB, especially in the cardiomyocyte. And next thing, we examine what all pathways have been impacted due to the loss of TFEB in the cardiomyocyte. So we performed transcriptome analysis in the cardiomyocyte of these mice, and we found that there were 1,600 differentially expressed genes, and out of these 1,600 differentially expressed genes, majority of the genes were involved in pathways which were related to cellular metabolism, apoptosis, cell death, lysosome autophagy, and lysosome function and cardiac function. And most importantly, among the genes involved in cellular metabolism, we found that 72% of those genes were involved in lipid metabolism pathways. So definitely we next want to assess the role of TFEB in regulating lipid metabolism in the cardiomyocyte. So indeed, we first examined the two important protein expression in the cardiomyocyte of these mice, that is PPAR alpha, which is important to regulate fatty acid oxidation, and CD36, which is a fatty acid transporter. And both of these protein were significantly decreased in TFEB knockout cardiomyocyte compared to wild type cardiomyocyte. And then we next also assess the ability of, when you alter TFEB levels, the ability of TFEB to form lipid droplet in the cardiomyocyte. So we used H9C2 cells, and when we overexpressed TFEB using adenoviruses, and subsequently we treated these cells with BSA control as well as with oleate to induce lipid droplet formation. So in M-cherry control group, compared to BSA, this is white bar, we can see that oleate is able to significantly increase the lipid droplet formation. And when we overexpress TFEB overexpression significantly reduced lipid droplet formation compared to its M-cherry control oleate group. And most importantly, it was very interesting to see that we also knocked down TFEB using shRNA. It was interesting to see that when TFEB was knocked down, it not only induces lipid droplet formation, but it also increases the lipid droplet size compared to its shRNA control group. So together, these data suggest that TFEB play a very important role in regulating lipid metabolism in the cardiomyocyte. And since we now know that TFEB is important in regulating this process in the heart, we lastly want to know that what is the implication of loss of TFEB in terms of cardiomyocyte function. And therefore we measured the cardiomyocyte contractility in TFEB knockout mice and wild type mice. So to examine TFEB cardiomyocyte contractility, we isolated cardiomyocyte, and then we placed this cardiomyocyte at different frequency ranging from 0.5 to 2 hertz. So we found that sarcomere length, SL represents sarcomere length. So sarcomere length during contraction and relaxation, we didn't see any change in the sarcomere length. However, importantly, we found that rate of velocity of both cardiomyocyte contraction and relaxation were significantly decreased in TFEB knockout cardiomyocyte compared to its wild type cardiomyocyte. And since calcium play a very important role in regulating cardiomyocyte contractility, we further measured calcium transient in the cardiomyocyte of these mice. And we found that diastolic calcium were significantly low in TFB knockout cardiomyocyte compared to wild type cardiomyocyte. However, the level of calcium during contraction, there was no change in calcium level during relaxation, during contraction. And most importantly, we also found that calcium transient was increased during contraction and calcium transient was decreased during relaxation. So together, this data suggests that despite low diastolic calcium and lower velocity of cardiomyocyte contraction, TFB knockout cardiomyocyte are still able to contract as good as wild type cardiomyocyte. However, these data were captured at baseline. If this is still going to be the case when we subject these mice to metabolic stress, such as feeding these mice with high-fat diet, we don't know at this point, and this is what we are planning to do it in our future studies. So in summary, we found that TFB play an important role in regulating lysosomal biogenesis and function, as well as cellular survival. And importantly, TFB is important in regulating lipid metabolism pathways in the cardiomyocyte. And also, it play a very important role in regulating cardiomyocyte contractility and myocyte contractile proteins, L-calcium signaling. So with that, I would like to acknowledge my lab, my supervisor, Dr. Thomas, our collaborator, IMPART team, my supervisory committee members, and most importantly, the funding agency. And lastly, I would like to also thank Cardiometabolism webinar series for giving me this wonderful opportunity to share my work today. Thank you so much, and I'll be happy to take any questions.
Thank you very much, Deepsika and Purvi, for these great talks. And you can all raise your hands and ask questions. Well, I have a question to start with to Deepsika. So have you considered the role of liver in these axes between adipose tissue and the cardiomyocyte in terms of the amino acid metabolism?
Yes, so that's a very good question. Actually, there have been a lot of studies that have been done, which has examined the role of the liver and muscle crosstalk. So previously, it was known that the concept of the field was that most of the keto acid is produced within the muscle, but the muscle cannot utilize the keto acid, so it shuttles it out to the liver. The liver, in turn, cannot make the keto acid, so it takes up the keto acids from once it is released from the muscle, and then it processes it. So that crosstalk is kind of established in the field, but now the radioisotope labeled studies and all the studies are done by Ibn Wang's group. They are showing that that is not the case. So muscles can in fact utilize the BCKAs, and livers can produce BCKA internally, which is like contrary to what the concept of the field was. So yes, the liver has an important role, but we did not study that. We could not, because all the tissues was procured during heart surgery, so we did not have any access to liver biopsy samples or anything like that. So, but that would be an interesting thing to study in our model.
Yeah, thank you. I have one more question before anybody asks. So Purvi, so have you looked into any markers of mitophagy because of TFB's involvement in mitobiogenesis and mitophagy regulation is really important for the cardiomyocyte?
I haven't looked into in this model, but we definitely, one of the preliminary study from in neonatal rat cardiomyocyte, where we actually, where I was actually knocking down just like doing some preliminary preliminary experiment to knock down TFEB and other members of TFE3, MITF family, that is TFE3. When I knocked down and as well as I treated the cells with other fatty acids, I found that Parkin and yeah, it was only, I think, Parkin were significantly decreased when we knocked down both of this protein. So that is the only preliminary study like I have done, but I haven't actually looked thoroughly in our model yet. But I'm pretty sure it has been known that it does regulate pink1 and Parkin protein. In TFB does regulate these proteins in other models.
So just to add on that, in our RNA-seq data, we have a mitophagy heat map actually that was not presented today, but in that heat map, we do see a couple of the targets of mitophagy that are altered, but surprisingly, in the TFB knockouts, we were hoping to see big changes in autophagy genes as opposed to other TFB models that is made in other tissues. In heart, we didn't see that. In fact, we see more of the changes in metabolism rather than in autophagy. So more of the canonical role of TFAB is not seen in the heart. It is more non-canonical. So we see more of metabolic changes rather than autophagy related changes. It was a surprise to us as well. So it looks like TFB has two different roles in the system, and one is autophagy related, but one is completely distinct from that. It is more regulating metabolism. So mitophagy changes were there few, but not as it is shown in other models of TFB deletion like liver or fat for that matter.
Thank you. So one more question from Lale. You have to unmute yourself, Lale. Oh, actually, your connection is not, you're not connected. Okay, now, if you can unmute yourself. Oh, we just lost her.
Can you hear me now?
Yes, great.
Okay, great. So I have a question to Purvi first. So in the beginning, you mentioned ER stress, right? Like that also has a role in glucolipotoxicity. Did you check any markers of ER stress? Because the reason I'm asking in this TFEB model, the reason I'm asking is because ER could be the link between, you know, the calcium metabolism as well. So what do you think? Did you check any ER stress markers?
We did check. We checked EIF2 alpha, both total and phosphorylation, and we did see that in response to glucolipotoxic stress, it's significantly reduced, especially in the treatment with palmitate and a combination of glucose palmitate. And we did also see that when you actually overexpress, it is able to increase the level back. So definitely we did see those changes. However, we haven't figured out the calcium part here yet, that like what calcium exactly does playing a role in our models, whether the calcium is coming from lysosome or the mitochondria or ER. So we don't know at this point, but we know from our transcriptome data that some of the proteins like calcium contractility and myofilament proteins were affected in our transcriptome, from our transcriptome data. And also, most importantly, some of the proteins play an important role in calcium cycling, such as SERCA and L-type calcium channel. Those have been significantly decreased from our transcriptome data. So some of the future studies we are planning to do is to perform the activity of these proteins and also to figure out what part of calcium is playing an important role in our model.
Okay, great. And I have one question for Deepsika. So you showed that insulin signaling is impaired, right, in hearts, cardiomyocytes treated with BCKAs, right? So, and then you showed AKT. Did you check the upstream signaling? In other cells, it's been shown that BCKAs regulate insulin signaling at the IRS level. Did you check whether it's at the IRS or IR or at the AKT level?
Yes, so we did. I didn't show the data here, but so we did the study both in skeletal muscle cells as well as cardiomyocytes. So most of the upstream signaling that I studied in the skeletal muscle cells, we did find changes with IRS signaling as well.
Okay. Yeah. All right. Great.
And Esther is asking a question.
I have a small question for Deficiency. Did you check any correlation with the microbiome, like with your BCKs and BCCHs?
Not in our model, but there have been studies that have shown that in obesity, the microbiome changes in mice, and they are able to influence the BCA catabolic enzymes. So there have been studies in the past, but in our model, we did not do any microbiome studies.
Okay. Yeah, thank you. Great story.
Thank you.
Well, if no last minute questions, this is it. Thank you so much for joining us.
Thank you.
And we'll see you next week.
Thank you so much, Bruin. Katie, thanks a lot.
Bye-bye. Thank you.
Bye-bye.
Thanks all the speakers.
Bye.
Bye.
Thank you so much to the speakers.